you very much. Hello, everybody. So today, the objective of my talk is to present some example of radionuclide measurements, combine mass spectrometry and radiometric techniques, and to discuss about difference and complementarities. So all of this work are performed in collaboration between two laboratories of the French Nuclear Agency, the CEA. The first one is the Laboratory of Analytical Development for Nuclear Isotopic and Elemental Analysis, which is called LANI. And the second one is the Henri Becquerel National Laboratory called NNHB. So these two laboratories collaborate since around 20 years and are located in the same place at Saclay in the south of Paris. So this slide is just a short presentation of the two labs, of their specificity and their analytical capabilities. So at LANI is a laboratory part of the Nuclear Energy Division of the CEA, and so the major mission of our lab is the determination of high precision isotopic and elemental analysis by mass spectrometry. And so we characterize high active samples like irradiated nuclear fuel samples, irradiated target or safeguard samples. And since 2014, we are part of the network of analytical laboratory from IEA for safeguard activities. So in terms of instrumentation, we have a large range of mass spectrometer instruments. And so, for example, TIMS, MCI-CPMS, or quadrupole ICP-MS, associated, of course, with separative techniques in order to purify elements before mass spec. LNHB is a national Henri Becquerel National Laboratory, is the laboratory of reference in France in the metrology of ionizing radiation, and it is a design institute for the LNE in France. And so is equipped of course of a large panel of primary techniques. And so we have in this slide just an illustration of several techniques that we have used in collaboration between the two labs. So it is, for example, Liquid scintillation counting with the 3:2 coincidence ratio or the 4p beta gamma coincidence method. So we have the major collaboration between the two labs concerns the determination of half-life from long life fission products, like fission products indicated in this table. So the collaboration are performed using internal funding or during international European projects like the Metrology for Radiation Waste Management European project for the determination of 3 half-life. Of course, we have other collaboration, like, for example, recently the determination of the absolute gamma ray emission intensity of the neodymium-147. And recently, we are associated in the European METRO-POEM project for the development and characterization of 2 reference materials. So my talk will be illustrated principally in the case of half-life determination in order to qualify with very high precision activity and mass. And so I will discuss about the recruitment necessary to attain this objective. So first of all, one very important step is the purification steps to isolate the element or the radionuclide of interest in order to prevent any spectral and non-spectral interference by mass spectrometry, or to prevent the presence of active radionuclides for radiometric measurements. And after separation, for mass spec, we use state-of-the-art mass spectrometric measurements by using isotope dilution associated with multi-collector mass spectrometer like TIMS or MCI-CPMS. So my talk will be illustrated with one example, which is the determination of the samarium-151 half-life. So in order to obtain the best precision and accuracy on the mass concentration of samarium, we must perform, first of all, a separation step. So in this case, the sample was an irradiated power of samarium oxide enriched in samarium-149, which has been irradiated in a French nuclear power plant. So after irradiation, we have around 400 microgram of samarium-151 present in solution. And as you can see in this table, we have a lot of isobaric interferences with europium and gadolinium isotope, and also presence of other radionuclides like europium-152, 54, or 55. So we must perform a purification with a high level for mass spec measurement and for radiometric measurement. So in the lab, we have developed several separation methods for fission products in nuclear fuel samples. And so in the case of lanthanides, we use high performance liquid chromatography using an acid, which is a hydroxyl methylbutyric acid, which is a very good complexant to lanthanides. And using this type of acid, we can separate with very high selectivity all of the lanthanide present in solution. So we can see in the left of my slide the separation of gadolinium, europium, and samarium in the targets. And we can see the very good selectivity of the separation. And so after purification, we can recuperate the samarium fraction. And we have checked by quadruple ICP-MS the purity of the fraction for mass spec measurements, and the LNHB has checked the purity for radiometric measurements. One of the major advantages of this separation is that the separation could be directly coupled with a quadruple ICP-MS in order to perform online measurement. We can see also different picture of the quadruple ICP-MS with a glove box around the source. So after separation, of course, we must perform mass spec measurements, and so we use the reference technique, which is isotope dilution, and one of the of a major requirement is the choice of the spike. We have not a lot of certified reference material certified in concentration and in isotope ratio for samarium, because it does not exist. So we have decided to choose a NIST SRM solution certified only in concentration and with an isotopic composition which is natural. So we have here a slide illustrating the isotopic composition of the spike in red and the isotopic composition of the sample in blue. So of course, in the spike, we don't have the presence of samarium-151. For isotope ratio, you have recommended value given by UPAC, but in this work, we have decided to qualify the NIST SRM solution by isotopic measurement using the thermal ionization mass spectrometry and using the method which is very used in the nuclear domain, which is the total evaporation method. The major advantage of this method is that we integrated the signal of all isotopes during all of the measurements, and so we have a minimization of the isotopic fractionation which occurs by teams. This is a method well used for qualification of uranium and plutonium isotopic standard in metrologic lab. So after the choice of the spike, we have sent this and purification of the solution. We have sent the solution to the LNHB, and the LNHB has performed the dilution. And after dilution, we have resaved the solution, and we have follow a metrologic approach in order to perform a mixture of sample and spike using 2 independent NIST SRM solution. And so we have performed 6 independent mixture, and the final results were the uncertainty of the final results was evaluated by combining all of the term of the equation of the isotopic dilution. And we have demonstrated that the main contribution of the uncertainty are related to the concentration and isotope ratio of the spike and to the isotope ratio of the mixing between spike and samples. So the mass concentration has only qualified in the lab in your laboratory, but for the activity measurements, the solution was sent to different European laboratories, and all of the measurements are performed by liquid scintillation counting. So you can see in this slide, and on the left, the value obtained by all of the lab during the European project. And so the final half-life was determined by the combination of the activity and the mass. So with this work, we have published a new value for the half-life of samarium-151 with a significant reduced uncertainty around 0.6%. And this work results in a new recommended value for the half-life of samarium-151 with a 10-fold reduced uncertainty compared to the previous one published in 2009. So this is an illustration of the collaboration between the two labs and the combination between mass spectrometry and radiometric technique. And thanks a lot for your attention.